I just wanted you to get the general idea that there is organization to the genome inside the nucleus, and it's a very important part of gene function, gene regulation, chromosome segregation, things like that. Also remember the study guides. Things that are in the study guides, look up. Okay. If there's something from the slides that I didn't cover in class, and if there is a question from the study guide, then look up in a reputable science source or one of the supplemental readings, and you should be able to find it, okay? Because I want to get into this topic today. Now, if you looked at the title of this topic on the class schedule, it didn't include aneuploidy, so I've included some things on aneuploidy here that I normally would have in a separate lecture, but got it all in together. Before we do that, though, you haven't seen this. It's been playing on a loop. This is time-lapse microscopy, fluorescence. The blue little blobs that are moving around are DNA. It's a fluorescent dye that labels only DNA. And this is a single cell. And oh goodness, it looks like some of the DNA is moving away from the big cell, and it looks like another smaller cell is forming. Anybody know what's going on?
Is it forming a polar body?
Yes. Do you know, let's see, I think you would be able to tell from this. Is this meiosis 1 or meiosis 2? Meiosis 1. Now you wouldn't necessarily be absolutely certain, but chances are if this were meiosis 2, you would have seen the first polar body here. Because remember, that's all held together, and this would be the second polar body. But this is a video that's available on a really useful website for anything having to do with human embryology. And I think I've got some links in the slides, and I'll point them out as I go along. And once I tell you, some of it will be review, but some of it might be new to you. I tell you more about meiosis, gametogenesis, I'll come back and show this again, because there's some really striking things that I want you to be aware of when you think about polar body formation. Okay, now this is where you should be, I hope, including, remembering some things, reviewing some things from Gene 3200, but perhaps a little bit more detail in a slightly different context. And aren't those beautiful? I mean, I love chromosomes, I love the cell, but I just think these are incredible. So there's the first polar body. This is after fertilization, though, so there's actually two. They're called pronuclei at this point. One of them, I don't think I know which one. One of them came from the sperm, one of them was from the oocyte. So one has a maternal genome, the other has a paternal genome. One of these is the second polar body. Does anybody remember? So this is completing meiosis 1. This is completing meiosis 2 when that second polar body is produced. What would have happened if this this is what's released at ovulation in a woman, right? Completed meiosis 1. What would have happened if this oocyte had not been fertilized? Would we see that second polar body? No, because this is something I'm going to review for you, is that it's a little bit unusual in female mammals. I don't know if it takes place in other organisms, but meiosis 2 is not completed until fertilization. So if that ovulated egg wasn't fertilized, it would just float along in the oviduct and the uterus and be lost. Okay, no point in going through meiosis 2 if it's not going to go anywhere. Right? Okay. Now you might be thinking, so what comes after this stage? So if you've got a fertilized egg, this is a zygote, when you've got this one cell and these two pronuclei, a one-cell embryo. What happens after this? Anybody know? Do we have more meiosis? Do we want more meiosis? Would this be considered to be a diploid? Yes. So what happens to this one cell? It goes through mitosis and becomes two cells. Each of those two daughters become, on. So we're going to be concentrating on, really, along with gametogenesis and everything having to do with it today. And then on Thursday, we'll consider essentially just the first week of embryonic development. Mainly what's happening with these what are called cleavage stage embryos up to the point of where they get implanted in the uterus. Okay, so pre-implantation development. I'll introduce you to a little bit of what happens right after implantation, but it's all to set you up to two things. One, I want you to know there's an incredible amount of things that are going on that fertilization triggers. You might just think, well, just the sperm enters the egg and, you get the two nuclei coming together and then you get mitosis, where there's a whole bunch of other things that are happening. They have to happen very accurately, right? Because this is a pretty crucial event for an individual. And there's a number of things, kind of concepts about pre-implantation development, what takes place after this, that are real important for your understanding of what pluripotent stem cells are. Okay, so that's all. Pluripotent stem cells are things that are taking place in the laboratory under defined conditions that really don't necessarily, the details of how their differentiation is controlled isn't really like what happens in vivo, but you have to understand what's happening in vivo to get a good appreciation of what's happening in vitro. Okay, so that's where I'm coming from with that. I'll show this again on Thursday, but I just want to give you some context here. This would be these stages. And don't worry, you don't have to learn any of this, but the staging, Carnegie stages, so there's a defined series of stages. Of course, any particular embryo or fetus will have slightly different time frame with, of course, with development. And these initial things are not to scale, but the zygote, so that's the two nuclei, right? That's what this looks like after one day, the embryo in relation to that. We're going to be talking about what's happening up until here, about up to here, okay? Now, actually, this is where we'll stop about here. I'll show you a little bit about what's happening here, but this is the main thing. This is actually, if you know a little bit about embryology, this is where gastrulation takes place, okay? What we'll talk about in a little bit more detail on Thursday is what's happening here with these cleavage stages. But before we get into the rest of this, and of course, there'd be a whole semester that you could take on human embryology if we were to get into the rest of this, okay? But where I want to start here is about here. Well, actually, yeah, between stage 12 and stage 13, because that's where gametogenesis starts. So remember, germ cells are a separate lineage from somatic cells. Somatic cells are all the other cells in the embryo or fetus or adult that are not germ cells. Okay. So here's the embryo, 4 weeks. Look at the size, it's really small. Here it is about a week later. Now the yolk sac is actually embryo derived, so that would be what's providing nutrition to the embryo. It's a little bit later, so if you don't recognize exactly what this is, there's what's going to form the head. There's where the heart is, as one of the first organs. It was the first organ to develop. This is the back, and these little segments here are called somites. That's the segmentation that is set up in your backbone. Okay. There is the forelimb. There is the hind limb. There is what would develop into a tail if humans had tails, but we do start out with what could become a tail. Now, this actually, amazingly enough, there's some differences in the head structure at this stage, but this looks very similar to a mouse embryo at the equivalent stage. Okay, so really, and any mammal that you would look at would look very similar. But what we're going to see, now it's hidden here. The yolk sac wasn't dissected away in this image. The yolk sac was dissected away in this image. And these are scanning electron micrographs, okay, so real high resolution. What we're going to look at a little bit is happening right about in here, where the genital ridge is located. The genital ridge is what will become the gonads, develop into the ovaries and testes, and it's right next to, and it's actually the developmental process is connected with what happens with development of the kidneys and the rest of the urinary tract. Okay, so it's often referred to as a genito-urinary tract. You don't have to know about that, but I just wanted to give you some context about that. That's actually, I shouldn't say, if I said that that's where the germ cells originated. That's where they end up. That's where the gonads are going to be developing genetic ridges. But what are called primordial germ cells actually arise earlier than that. And they actually arise outside of the cells, the structure that's actually going to be formed or is the embryo complex. Okay. And there's actually 4 stages. Now I took this right out of that chapter 1 from Carlson, so I do want you to understand this. Because it's not so much the origin and migration that I want you to understand. It's an interesting developmental process. But I really want you to get the appreciation that mitosis is very important to germ cell development. Right? Because you can have mutations that take place during mitosis, right? And of course, we're thinking most of the time about meiosis when we're talking about germ cells, but mitosis is very important. The other thing I want you to realize, and we'll come back to this, okay, we got the origin and migration, you'll see that in a second. Once those primordial germ cells they have to migrate through the embryo, and then they reach what will become the gonads, they reach the genital ridges. Well, a really interesting thing happens. They've been going through mitosis while they were migrating from their site of origin to where their destination is in the genital ridge. But once they reach the genital ridge, they stop. They arrest in the cell cycle. And basically they sit there relatively quiescent until, in terms of what would be happening in terms of chromosome segregation, they stay that way until either after birth or at puberty. The other interesting thing is, you might know this, in the male gonad, these PGCs, once they reach that genital ridge in the gonad, they arrest in mitosis. Female, the germ cells arrest in meiosis. And they never again go through mitosis. And so the number of germ cells that a female mammal, whether it's a mouse or human, the number of germ cells in that newborn female baby, that's the maximum number she's ever going to have in the back. The large number of them, vast majority of them, just naturally degenerate. The other thing, does anybody remember at what stage of meiosis these PGCs in the female gonad are arrested? So meiosis 1, meiosis 2, any particular stage of those? You remember?
Phase 2.
No. Prophase 1. Prophase of meiosis 1. What takes place in prophase of meiosis 1 is special. What? Crossing over, recombination. And remember, if you're talking about a female, a human embryo, female embryo, her oocytes arrest in meiosis 1, in prophase of meiosis 1. Remember when they're held together in that synaptonemal complex? We'll review that, right? For how long? Decades. Till puberty, then 1 oocyte per month, the menstrual cycle, 1 oocyte per month, continues to grow, gets ovulated, completes meiosis 1, and is released. So that's once a month until menopause at 40 to 50. So that complex of proteins that are holding together those chromosomes in the germ cells of the female, they have to be maintained for literally decades. That has some consequences that I'm sure and significant that we'll talk about as we do that. Okay. So let me just show you a little bit about the origin and migration. We won't talk any more about mitosis, and then we'll get right into meiosis. You don't have to worry about this. I mean, I want you to know that there is this structural and functional maturation, and you can read about it in that chapter, but other than knowing that there's a stage, you don't have to know the details of it. I just want you to know. But that's also a very interesting process that's going on. Okay. So now we're looking at the side of the embryo. this is about 4 weeks to 6 weeks, okay? So there's the head, there's what would become the tail. Those little red cells there, they're actually literally when they're first designated, they're probably just less than a dozen, like 8 to 12 or something like that. And they actually receive signals from both the embryo and this structure called the allantois, which is what forms the umbilical cord, okay? But what's happening here is this is before the gut has formed, but what happens is those little cells along here will just kind of invaginate into this area and forming the tube, which is going to become the hindgut. So we've got a tube inside us, right? That really goes on from our posterior to our lung. Now there's a lot of other things that are happening in that, but it's a tube within it. And these germ cells are just kind of carried along in as this hindgut is invaginating and forming. And they're just kind of carried along passively. No, they do start migrating. Now, if you take this embryo, so it's laying like that, and then you kind of turn it slightly to the side and then cut right through there. That's what this section is showing. Now, so this part, this is the head would become curling up over here. The tail part would be curling down here. Now we've got the gut formed. We don't have any organs yet, other than the gut. There's no differentiation, specialization going on. But there's this structure, essentially like some membranes, that have attached the gut to the body wall. Well, at a particular stage of development, the primordial germ cells will leave the gut and they'll start migrating towards the genital ridge. And the genital ridge isn't even completely formed at this point. It's kind of forming as this migration is taking place. And remember, they're still going through mitosis. Now, if you go a little bit later, now we've turned this embryo from being like that to making a nice cross section. Now we're seeing the gonads, what we call the genital ridge. Here's that membrane structure, the dorsal mesentery. There's the gut that the cells are migrating. We're not seeing the other side of it. These are all in mitosis. They're expanding, I forgot the number, maybe 1,000-fold, if not more, 100,000-fold. I should remember, but I don't. But then when they're continuing on mitosis, shortly after they reach here, they get signals from these cells that are in the developing gonad that tell the cells to arrest in the cell cycle. Now, up to this point, there's no genetic or morphological difference between an XX and an XY embryo. They look exactly the same. In fact, they have the potential to develop to either sex. Right around the time when migration of the PGC, so the gonad is complete, sexual differentiation occurs. An XY embryo, there's a particular gene will get transcriptionally active. It's the male sex determining gene. Anybody remember what that's called? SRY, yes. Obviously not present in XX individuals, so you don't get male differentiation. So now we've got an embryo. At the 6th week of development, we're seeing the whole thing. This is the genital urinary tract. We're not even seeing the gut anymore. But there is the genital ridge. It looks like a little hot dog. And actually, if you stained these for, there's actually a nice red pigmented staining that you can do for primordial germ cells. It would just look like a hot dog there with lots of red dots in it. It's kind of cool. Now these, the mesonephros and paramesonephros, you don't have to know those, but just in case you're wondering, both of these sets of ducts form an undifferentiated embryo. In a female, the mesonephros degenerates and the paramesonephric duct develops into the oviduct. In a male, the paramesonephric duct degenerates and the mesonephros develops into the seminal vesicle, urethra, all those other things that males have. Okay. Again, you don't have to know that detail. Meanwhile, SRY has been activated transcriptionally only in the genital ridge. And even though this has been identified as the male, the master regulator of male sex determination in mammals, it's still not clearly known, as far as I understand, what is activating expression of SRY. We know that once SRY is being transcribed, what it does is activate the transcription of a number of genes downstream to it. And one of the most important of those you don't have to know of, but that is well known, SOX9. Then SOX9 is another transcriptional regulator that activates a whole bunch of other genes. So of course, remember, this is only happening in males. It wouldn't be happening in females. Okay. So this is just to show you what's happening. Now if you take a lateral view of the genital ridge, you can stain red for the germ cells, green for the somatic cells. Now a lot of times I'll be showing you fluorescent images of cells, tissues, stuff like that. Always pay attention to the color code, and I won't necessarily always tell you what the protein or macromolecule that's being stained is, but if I just tell you what the cells are, it's color coded. Does anybody have red-green color blindness? Any of you guys? Good. Because unfortunately, red and green are the most commonly used fluorescent colors. Unfortunately, that must be kind of tough for somebody. The other thing that's happening once SRY gets expressed in the XY gonad is that these tubules start forming. The germ cells start organizing themselves inside these little tubules. Those little tubules are what are going to form the seminiferous epithelium that continue in the adult testes. Okay? Female doesn't have that kind of organization. Okay, now this should be review. I'm not going to go through. I'm not going to review all of it for you, but this is meiosis. Meiosis 1, meiosis 2. Review all of this, think about what's happening at the level of the chromosomes. In particular, prophase 1, 2, what does C mean? What does 4 C? Does that mean it's tetraploid? Is it tetraploid? No, it's not tetraploid. In fact, this is the correct nomenclature. Unfortunately, a lot of times you'll see prophase 1 in textbooks labeled as being 4N. Technically, that's not correct. 4N would indicate that there's 4 sets of chromosomes, and they're not 4 sets of chromosomes. This is still a diploid, even though the amount of DNA is doubled. Because remember, at this point, every one of the chromosomes consists of a pair, a sister, of chromatids. That's still only one chromosome. And it's considered one chromosome until they separate. In meiosis, they separated anaphase 2. Okay. So that 4C means it's got 4 times the C value, which is a haploid value of DNA, but they're still diploid. Make sure you review, I'll go through some of the things with cytosine, actine, diploidine, diakinesis. Now, prophase 2, that is the reductional division. The homogenes are separated. The gametes, of course, are haploid. And as we saw on the animation, and I hope you remember, in a male, there would be 4 haploid gametes being formed. Females, do we get all 4 of these? Sometimes we could get all 4 of them, but because of the asymmetric division that's resulting in those polar bodies, we still do the chromosome segregation in meiosis 1 and meiosis 2, just like we would. What's different is that instead of cytokinesis taking place at the end of meiosis 1 and then in meiosis 2, that essentially in the male, you get 2 daughter cells that have equivalent cytoplasm. Because of the polar bodies and because this is nocturnal, there's asymmetric division. But what this does is a way of having all of the, or most of the cytoplasm being present in the oocyte, not in the polar bodies. So you can think of the polar bodies as just being contained in the DNA, the chromosomes that are segregated. There's a little bit of cytoplasm there. Okay. But in terms of probability, let's say the probability of this particular cell being the oocyte and one of these being the first and second polar bodies, that's still 25%, just like you would expect. Okay. Oh, and as we review, recombination takes place. Now, Kaijan is going to tell you more later in the semester about what's happening at the level of sequence analysis with recombination. I just want to review something about recombination. Of course, it's one of the ways that genetic variation is created, so it's absolutely essential. But it's also, if you don't have recombination, meiosis will be arrested. So in a sense, recombination is also essential for fertility. Okay, and we know this for sure from mouse models, where they've been mutated for genes that are essential for recombination. They arrest in meiosis. Males and females. Some are sex-specific, others affect both sexes, but we know for sure. Okay. And so there has to be at least one recombination event per chromosome. And it's also important, and again, there's direct evidence from the mouse, there's inferential evidence from human, that the position of the crossover is important also. Again, this is a detail you don't need to know, but it's just something to be aware of. And if you're interested, explore it further. Now, you know about crossover. That's the physical exchange of DNA, right? Reciprocal recombination. Think Holiday junctions, double-strand break model of recombination. But keep in mind, we can also have non-crossover events that are essentially where the double-strand break model, double-strand break recombination has been initiated, but it doesn't resolve itself in a crossover. Because remember, you're thinking of the crossovers is a physical exchange of DNA between non-sisters. But you can have gene conversion. So imagine, 4 chromatids. You can actually get conversion of one of those to the allele of the other. So you can actually end up getting 3 chromosomes in meiosis that have identical alleles, and the 4th one, I'm talking about in a heterozygote. Okay. I don't know if Kaijan will talk about gene conversion more later on, but I just wanted to point out that it can happen. Okay, now I do want you to review this. I do want you to learn the names of these phases, or subphases of prophase 1 of meiosis, because a large part of the reason I want you to learn this terminology is just a way of remembering the very defined series of events that have to take place. Leptotene, this is where the synaptonemal complex has these proteins that hold both the sister chromatids and the homologous chromosomes together. We have to have that structure formed. It's complete synapses by zygotene, the zipping up of the homologous chromosomes. Pachytene, this is where recombination actually takes place. Okay, and so in terms of the process of recombination going on, this is the most important stage. Then diplotene, this is where recombination has resolved. Those Holiday junctions would have resolved, and you'd either have crossover or not crossover. Now, at the diplotene, these homologs are still held together at the chiasmata. That's a physical structure that you can see in the microscope, under the microscope. But they do start separating. Diaphonesis, they just kind of extend that separation. Now remember, this is prophase 1. What comes after prophase?
Metaphase.
Metaphase. So these chromosomes have to start pulling apart. They actually get a little bit more condensed too. So they're kind of pulling apart and getting shorter to allow them to line up. Now I do want you to go, there's just the first part of this, I want you to learn the important components here. Like SPO11, so here we've got a pair of sister chromatids. Here we've got a pair of sister chromatids. Okay. SPO11 is the enzyme that makes that double-strand break in one of those DNA, one of those chromatids, just one of them. Now, what you're taught and what you can think of in a general sense is that double-strand break made by SPO11 could take place anywhere along the chromosome, each of the chromosomes. In reality, there are hot spots, and that's something that Kaijan will talk about later on. But for right now, just imagine it taking place anywhere. We've got the synaptonemyl complex, that's what these red and blue lines are starting to form. And cohesins, you may have heard of those. Those are proteins that are involved with holding together both the sister chromatids, and they're actually important for holding together the homologs too. Once SCO11 has made that double strand break, this protein, called that funny symbol, gamma, that means it's a phosphorylated protein, phosphorylated H2AX. Anybody want to take a guess of what H2AX might be? You might not have heard of H2AX. Anybody heard of H2A? It's a histone. It's one of the canonical histones that forms the, right, there's H1, that's not part of the nucleosome, but H2A, H2B, H3, and H4. Well, I don't know the details of exactly what it does, but phosphorylated H2AX, X means just it's a variant. I think it only differs by a couple amino acids from the canonical, the normal H2A that's in the nucleosome. But that's phosphorylated, and that localizes right next to the break. And then you get these two proteins coming in. You don't have to know the details of these, BMC1, RAD51, but these are what are mediating. If you remember that double strand break repair model, right, you've got strand invasion, where one of the broken strands invades the non-sister, and then you've got DNA. That's all being mediated by these two proteins. Okay, so I do want you to know this because this relates to something that will be discussed later. That was all in leptotene, early prophase 1. Zygotene, zygonema, we get the complete synaptonemyl complex. Now we get the homologs that are starting to line up together, zip up together. Now paclitine, again, you don't worry about what these proteins are doing, MLH1 and MLH3, but they're part of what you could call a recombination nodule. This is where the latter part of double-strand break recombination and resolution is taking place. And so you could have a series of, you'll see a picture in a minute, with a series of several nodules that contain these and other enzymes are located along each of them. Then by diplotene, you still get this physical structure that represents the site of the initial recombination event, and you start getting the homologs pulling apart a little bit. And just to show you some pictures, these are actually, I think these are from sperm. I'm not sure, the spermatozoa, I'm not sure if these are oocytes or males. But anyway, just to show you now, again, the color coding. SYCP3 is one of the components of the synaptonemyl complex that's staining red. And then the green staining in each of these panels are different proteins that mediate these processes. So you can get a good idea here with the red staining for the synaptonemyl complex and zygonema. You don't have the complete complex forming. You don't even have complete chromosome condensation taking place. It's in progress. By paclitene cells, you've got the homologous pair of chromosomes. Remember, each of these, what appears to be one structure, consists of that tightly synapsed homologous pair of chromosomes. The maternal and paternal chromosomes for each pair are lined up together, and remember, each of those chromosomes consists of a pair of sister chromatids. So there's actually 4 strands of DNA here. But this staining, these are showing the recombination nodules that contain MLH1 in this case and other enzymes. Now you no longer have any recombination nodules, and you can also see that the homologs are starting to separate. So I hope this lets you, being able to picture this lets you remember the events that are taking place and then just gain those names. This is a male. That's the sex chromosome. That's an X and a Y chromosome. I won't go into why that's got a lot of phosphorylated H2S on it, but yeah. And this is just another example. Now we've got, what are kinetochores? Blue staining for kinetochores. So there's just one little circle on each of the chromosomes for the kinetochore. What's a kinetochore? That kinetochore, remember, attaches to the centromere, and it's a spindle fibrils that actually attach to the kinetochore that is attached to the centromere. So you can actually, in the old days, now these are actual human fetal oocytes, you can collect, there have been some studies, this one was done in 2009, where you can collect human fetal oocytes. Okay, now, the woman would actually have to be donating them, and this also, sometimes this is the case of elective surgery, when a woman is willing to donate them. But you can, in the old days, this is, you can get an estimation of recombination frequency and the location by just counting up and scoring the position and number of the recombination numbers. Now this is showing you reviewing meiosis. Now I do want you to know these terms in the male and the female. This is just another representation. This is from the supplemental chapter telling you the nomenclature for each of these stages. It's 1/4 after. Why don't we take like a 5-minute break and then we'll continue.